Hello everyone, my name is Donghyeon Lee and I'm the CTO of Celloid. First of all, I'd like to thank you for the kind invitation. It's truly an honor to have this opportunity to connect with such outstanding researchers like you. I'm currently in the flight toward the US, so I regret that I couldn't meet you in person. Today, I'd like to introduce Celloid's products and technologies that enable scalable and reproducible organoid production. Before we get into the details, let me first briefly introduce who we are at Celloid. Celloid is a company founded by mechanical engineers, combining expertise in electrospinning, the material processing, and 3D fabrication, and robotics. Based on these engineering technologies, we provide solutions for reproducible and controllable 3D cell culture. Currently, we offer two main products, the Nestwell, a non-fibrous membrane-based permeable microwave array, and OrgaNest, an automated 3D cell culture system. I will explain both of these in more detail later in this presentation. The main challenge we aim to address begins with the standardization of organoid culture. As you know well, organoids have great potential, from drug screening to regenerative medicine, but culturing them remains very difficult. This lack of standardization have become a major obstacle to the industrial application of organoids. Even the OECD has emphasized that in order to use organoid-based assay as an animal alternative testing method, it is essential to ensure what they call the 2R1T principle: reliability, relevance, and transportability. From an organoid culture point of view, reliability refers to reproducibility and repeatability of the result, and relevance corresponds to how closely the organoid mimics the structure and function of the human organ. In other words, its maturity. And transferability represents how easily the culture process can be replicated by others. The key problem is that most of the widely used organoid culture methods fail to satisfy both reliability and relevance at the same time. Actually, there are tons of 3D cell culture methods proposed in academia, but let me focus on the two most common approaches in this slide. First one is the use of hydrogels such as Matrigel, and the second one is the use of impermeable plastic microarrays such as Agribel. The hydrogel-based method provides an environment that mimics the native extracellular matrix, allowing efficient exchange of nutrients and growth factor and proper removal of waste. This leads to high biological relevance of the resulting organoids. However, it is almost impossible to control the size and functionality of functional uniformity, making it difficult to ensure reliability or reproducibility. On the other hand, in the case of impermeable microwave arrays like Agribel, the number of cells seeded per well is fixed within confined geometry, which helps achieve high uniformity in organoid size. However, since the bottom of surface bottom surface of the wall is non-permeable, nutrient and waste exchange is limited, and it results in lower cell functionality and reduced relevance. In addition, organoid culture requires a high level of technical expertise, mainly because the culture periods are extremely long and organoids are quite tricky to handle. If we look into various organoid culture protocols, some can be completed in months, but for more complex organoids, such as brain organoids, the culture period can be extended to nearly a year. During such a long-term manual handling, the risk of contamination and organoid loss can becomes very high. Ultimately, these losses turns into significant increases in production cost. To address these challenges and enable reproducible production of organoids with high uniformity and maturity, Celloid provides two complementary solutions. The first is a nanofibrous membrane-based permeable microarray named Nestwell. Uh, like conventional microarrays such as Agrewell, it confines a defined number of cells within each microwell, allowing organoids to maintain consistent size and function throughout the culture period. However, unlike traditional microwell arrays, the sidewall of each microwell are made of permeable nanofibrous membrane, which enables efficient exchange of nutrient, growth factors, and waste, thereby supporting both uniformity and physiological relevance. The second solution is the OrgaNest system, an automated perfusion-based platform for 3D cell culture. Unlike pipetting-based medium exchange, which requires opening the culture chamber and which increases contamination risk, OrgaNest system performs automatic media exchange through closed-loop tubing, shown here, allowing slow and gentle perfusion that minimize the organoid loss and contamination. Additionally, the system is equipped with the motorized microscope, enabling users to monitor organoid growth and morphology change without disturbing the culture environment. As shown in this illustration, Nestwell and OrgaNest can be used in combination, providing a fully integrated workflow from uniform organoid formation to long-term automated culture and monitoring. I will explain the details, functions, and use case of these two solutions in the following slides. For more details, let me start with the Nestwell platform first. As mentioned earlier, Nestwell was developed to enable the production of uniform and mature organoids. Its precisely engineered 3D-dimensional microwell geometry allows multiple organoids to be cultured uniformly within a single insert. Moreover, since each microwell wall is made of highly permeable nanofibrous membrane, soluble factors in the media can diffuse efficiently, providing the organoid with a stable and nutrient-rich environment. This permeability plays a critical role in organoid differentiation, and to verify its impact, we conducted both simulation studies and kidney organoid culture under different membrane conditions. By intentionally reducing the porosity of the membrane through controlling thermal treatment, we observed in simulation that nutrient diffusion was significantly impaired as permeability decreases, and consistently in the actual culture experiments of kidney organoids, the organoids grown on the less permeable membranes exhibited reduced gene expression levels, demonstrating that the membrane permeability directly affects the organoid maturation. Our first proof of concept study using the Nestwell platform was performed with kidney organoid. As I've emphasized several times, compared to the conventional hydrogel-based culture, Nestwell enabled the generation of organoids with remarkably uniform size. In addition, by simply adjusting the microwell dimensions, we were able to precisely control the final organoid size according to the desired specification. One more unique feature of Nestwell is that organoids are suspended in the middle of each microwell, supported by the permeable nanofiber membrane. And because of this configuration, the media beneath the membrane can be completely removed and replaced without disturbing the organoid themselves. This feature not only minimizes the organoid loss, but also allows for precise and complete media composition change during during complex or following complex differentiation protocols, reducing carryover effect from residual medium. Also, when we perform the whole mRNA sequencing on the kidney organoid culture in Nestwell, we found that their cell type composition was more similar to human and mouse kidney tissues compared to those cultured in impermeable microwave array, that, in which is well. Even more interestingly, the organoids exhibited signs of spontaneous vascularization, indicating enhanced physiological relevance achieved through our platform, the Nestwell. Organoid culture with high uniformity and maturity, as achieved with Nestwell, enable more precise and reliable drug evaluation. As a proof of concept, we induced polycystic kidney disease, also called PKD, in kidney organoids, and then we treated them with a CFTR inhibitor to monitor the therapeutic response. In the organoid culture in hydrogel layers such as Metrogel or in an impermeable microwave array like Agribel, the large variability in organoid and maturity made it difficult to observe any statistically significant differences across drug doses. In contrast, organoid culture in Nestwell showed a clear dose-dependent inhibition of cyst formation as the concentration of CFTR inhibitor increases. This demonstrates that improved uniformity and maturity directly enhance the sensitivity and reproducibility of organoid-based drug testing. In a similar approach, we recently cultured liver organoids using Nestwell platform and conducted a proof of concept study by treating them with acetaminophen. Although this experiment is still in progress and we haven't completed the quantitative analysis yet, we clearly observed that dose-dependent response as shown here. Higher concentration of acetaminophen led to noticeable shrinkage of organoids. These preliminary results suggest that Nestwell can also be effectively applied to liver toxicity and drug response studies, further demonstrating its potential as a versatile platform for organoid-based drug screening. Now let's move on to the OrgaNest system, which is our automated 3D cell culture system designed for long-term organoid culture. The OrgaNest Basic Plus system is composed with three main modules: supply, perfusion, and vision. The supply module stores multiple types of media under refrigerated condition and includes an integrated pump system that delivers or drains the media through tubing, shown here, connected to the culture chamber. The refrigeration function prevents media degradation during prolonged experiments. Also, because the media is delivered very slowly through this thin tubing, it is gradually warmed to the appropriate temperature before reaching the organoid. The perfusion module contains the culture chamber itself and incorporates with multiple valves that precisely control the direction and timing of media flow. Finally, the vision module is equipped with a motorized microscope, allowing users to monitor organoid growth and morphology change in real time without opening the culture chamber. Our OrgaNest Basic Plus modules are designed to be very compact, so the entire system, except the supply module, can be installed inside the standard suit incubator. And this modular design also enables flexible configuration, and to suit various laboratory environments and experimental needs of many researchers. Also, one of the key features of OrgaNest is its perfusion-based media exchange mechanism. And because the media is supplied and removed through tubing in a closed culture environment, the system can minimize contamination risk throughout long-term culture. Moreover, the extremely low flow rate ensures that organoids are not exposed to shear stress and can grow stably and undisturbed over extended culture period. Another key feature of the organoid system is that it operates not as a multi-use bioreactor, but in combination with a single-use cartridge, which is disposable. As I mentioned earlier, organoid culture is a long-term process, which makes it highly susceptible to contamination. Once any part of equipment comes into contact with the culture media, it becomes extremely difficult to maintain a sterile environment over several weeks of operation. To address this, the OrgaNest system utilized a detachable and disposable cartridge that serves as a sterile interface between the culture and the device. Each cartridge contains 5 media bottles and 1 waste bottle, along with 6 independently controlled culture chambers. The cartridges are provided pre-sterilized and vacuum sealed, so users simply unpack the cartridge, load the media and cell into the cartridge, and attach it or install it into the system. Uh, then, after the user set up the desired culture protocol, then the OrgaNest system automatically performs media exchange and perfusion the media throughout the entire culture process. Also monitor the change of the organoid inside the chamber. Currently, each of the 6 culture chambers in the OrgaNest system, especially in the cartridge, can be equipped with either celloids, nest-well inserts, or be applied to the hydrogel domes like Metrogel, depending on the user preference. For users who aim for uniform organoid production, we recommend the configuration shown on the left using nest-well. For those who wish to automate their existing manual culture protocols more directly or seamlessly, the configuration on the right using the Metrogel dome can be selected instead. The video shown here are time-lapse recordings of organoids being cultured in both configuration, nest-well and Metrogel domes, and monitored through the built-in microscope, the vision module of the OrgaNest system. In addition to automating traditional manual procedures, the OrgaNest can also features a gentle mixing mechanism that periodically withdraws, drains, and re-injects the media within each culture chamber. This process helps create a more homogeneous culture environment compared to the conventional static culture conditions. So far, Celloid has dedicated to developing technologies that enable reproducible and controllable organoid culture. As I introduced earlier, we have launched 2 key products, Nestwell and OrgaNest, which are designed to make organoid research more standardized, scalable, and efficient. With these solutions, we aim to support a wide range of applications from basic research and drug screening and large-scale organoid production. If you are interested in our products or potential collaboration, please feel free to contact us anytime. we would be more happy than to explore opportunities to work together with you. This is the end of presentation, and since this presentation is being delivered through a recorded video, unfortunately I won't be able to take questions in real time, so please feel free to reach out to me by email at any time. Then I will be happy to respond and continue to discussion with you. Thank you for very much. Thank you very much for your time, and thank you for watching this presentation. Thank you.